This is an important session for us to gather information from you. We will give you a brief summary of some of the high points that we think are important for HIBALL going forward. But Katrin will talk for about 10-15 minutes, I will talk for about 10-15 minutes, and then we would like to hear from you. So I have seeded a few questions with people that who I think have important perspectives and will help us to guide the further development of HIBALL over the next 5 years.
Yeah, so let me start with some thoughts, and it's, so to say, it's about Atlas. It's also about what is an environment to continue research, and perhaps from there really also to develop together some ideas here with this room. So I would like to start with the notion that BigBrain was a starting point for many types of research. And it was also a very big push, I would say, from the BigBrain to develop a comprehensive atlas and develop therefrom next generation AI. And we did it in the Human Brain Project between 2013 and 2020. And the result of this development was a digital platform, a research infrastructure that is a European one, and that provides data, tools, and services and access to atlases. And for the atlas, it was really a very important collaboration with the colleagues from all over Europe, because it gives us the possibility to embed the atlas into other activities. And we heard today also in particular in the last session how important it is not only to have methods, but also to make them usable, to scale them up, and to link them with other types of data. And that was a little bit also the motivation of this eBrains infrastructure. And eBrains is also, I would say, a putative or a perspective because Europe and Canada, last but not least, have concluded a contract of collaboration. And while earlier funding activities in Europe were really limited and restricted to Europe, at least we have now the possibility to have partners from Canada, also from UK, also from Switzerland now again, Australia I think again, as beneficiaries in normal European projects. And I think that's something to think about in this respect. And I mean, this environment that we have built in the eBrains research infrastructure also includes the atlas, and you heard yesterday about the Zebra toolbox that was developed mainly by Timo Dickscheid and his team. And the Zebra toolbox really is a Python-based environment to build workflows in a very easy way, crossing the different spatial scales and modalities. And this is so important because the brain is a complex system. We heard very often during the last 2 days about multimodality, multi-scale, multi-level, and to have a software tool suite that is allowing to jump between the different spatial scales and modalities is extremely important. And it starts with an easy online viewer that is intuitive and then goes to a Python interface. Using this Python interface, really with a few lines of code, you can extract quite complex data. And this opportunity is used by our common friend Victor Girza in Marseille, who is developing with colleagues worldwide, with René McIntosh, also with Petra Ritter, the virtual brain. And this is a simulation modeling and simulation framework that he has applied in particular to predict the outcome of surgery in epilepsy patients. And he is developing personalized models of these patient brains that are being informed by IEG data, DTI data, but also by atlas data, and you can see here easily there's a BigBrain. So that's a very concrete use case where we take advantage of the BigBrain in order then also to develop these individualized models, and I think it's one of the rare cases where a clinical trial is ongoing still. The analyses are not yet finished, hopefully next year. And then we hope really that we can provide some progress, some more benefit for these particular patients. So multimodality is really an essence, and it is everywhere, I would say. Also in collaboration with our colleagues in Padua, and we just heard one talk in the last session. We are developing in the Human Brain Project, based on the atlas, based on the BigBrain, a possibility to superimpose multimodal, pentamodal imaging data of patients across entire Europe. And eBrains is providing a framework where you can get access to this data, but where you have also rather easy to go tool to compare the different maps. And again, in the last session, we saw different approaches how to compare maps, and here will also based on Zebra Python very soon will be a software available to do such type of analysis with the data. And some of us are working in hospitals, and this is always, I would say, a challenging question. Yeah, you have firewalls, you work really behind this firewall, so how to use such infrastructure like eBrains. And Timo here together with Rainer Göbel from Maastricht, they are developing an fMRI analysis tools, and that's a little bit a successor, I would say, of the anatomy toolbox, of the SPM anatomy toolbox that Simon Eickhoff developed more than 20 years ago now. So this is the next phase, certainly, and it allows in an easygoing way to download the tool, make it running on your laptop, and then do structure, functional analysis in a very easygoing way. We also see, of course, that there are worldwide different applications and software tool suites, and some of them developed over many, many years, over decades, and they have their users, they have their preferences, their advantages, sometimes also disadvantages. So what we are now trying to do is really also to support these existing software packages, and AFNI is one out of it, and the Jülich Brain Atlas is now the default setting in this AFNI Atlas, and those of you who use AFNI, they can then have a nice and a more easy access to the Jülich Brain. And with Douglas Grave and Bruce Fischel at Boston Harvard, we are developing possibilities to extract surfaces of our postmortem brains, including the BigBrain. And these surfaces are then be projected to the surface. And this is a tricky and very difficult task, I would say, because the data on which the Jülich Brain cytoarchitectonic maps are relying, they are 20, 30 years old partly, and MR imaging was not at the same level. Yeah? So they have quite a number of problems, and to extract surfaces with high precision is really handwork, and a lot of manual interaction is necessary, and quality control. But I'm confident that this can work. And then, of course, we will have the possibility to map the maximum probability maps or the individual maps to individual patients, also to predict findings in vivo imaging studies. And I would like to emphasize that the combination that we have with the BigBrain and the cytoarchitectonic probability maps, this is a very important combination, and this is unique worldwide. So the BigBrain is so important because it has this very high spatial resolution. And you can start to extract cellular parameters based on the big brain and use layers, cortical areas, and other parameters. The cytoarchitectonic probabilistic maps, they are important because they are based on the mapping of 10 brains. So they include from the very beginning information about intersubject variations, and we discussed it during the past 2 days that this is an important aspect. But it is more, it gives you, so to say, a region of interest where you can then look into other data resources, and based on this microstructural data, you can extract sound biological information from any other maps. And I think this coherence and also the, so to say, the approach that we use, the same method in the big brain, but also in cytoarchitectonic probabilistic maps, this is of course much more than just a label that you can put to any map. You can use any labels, of course. But the question is, what is the biologically most sound label? Where do you have, so to say, the highest precision also in extracting other information? And for the big brain, we have anchored some time ago also the 1 micrometer sections, and the first big brain cannot be processed in the entire extent of the 7,404 sections. The quality is not good enough of the histological sections, and they also became older. So that's not possible, but what we are providing is across the entire brain having these high resolution 1 micron sections in the big brain. And this then allows us also to identify individual cells, and you heard about our new algorithm that is able on the fly to detect cells, and you can then extract any parameters from this histological section. But it's also clear we want to go 3D. Yeah? So at the end, when we want really to get a comprehensive understanding of the human brain, that means that we have to bridge the macroscopical or mesoscopical scale, the scale where you see imaging data or where you have PET data or diffusion data. You need somehow to relate it to the cellular level, because cells are the information units that process information. So we want to go to 3D. And this is done by Marcel in our group. And the big challenge is that when you cut entire human brains, you also cut cells, you also cut blood vessels. And when you then try to stack them and to register them to each other, then it becomes clear that you really have to go to the cellular level in order to achieve a good registration. And when you just do it section by section, then of course you buy in quite some number of artifacts, the banana effects that is very well known. So it is important to combine local registrations also with more global registration, and this is what Marcel is doing. And he is using, I think, a very interesting approach that will result in larger and larger regions of interest. So 3 we have already provided to the community of a few cubic centimeter. And this is, so to say, the first time we are also presenting big brain number 3. So this is a brain that we have stained for chrysal violet. And that contains 1 micron isotropic data. It is not yet corrected for staining. This will be the next step. We have partly a cell-on cell precision, and you heard about Tiamet integration by Alexander Oberstraß. So this is a little bit the future perspective we would like to go to higher spatial resolution, and then to also provide this as a resource to the community, so that we really can image at the cellular level all the details, and using AI, of course, also to predict. So this is 1 cell type, if you like. These are neurons mostly, and nuclei of glial cells. And of course, we are very much interested in better understanding the cell types. This can be done with immunohistology. And Roxana Koymans from Amsterdam is staining here large sections of the human brain with different immunohistological targets. And the special thing about this image is that she is using triple staining with immunohistology, so she is doing free-floating staining, which is absolutely difficult for such type of sections, but the sections have also the fiber architecture based on polarized light imaging. So all the modalities come together, and that's a little bit, I would say, the dream from the very beginning of the past century of Oskar Vogt. So by having this polarized light imaging and making the bridge to diffusion imaging, as done here by our French colleague in Saclay, by Cyril Popin and Jeff Mangin, we are really capable to visualize single axons and thin fibers. And when you are moving across the brain, I think you immediately see that there are rather sharp changes of the pattern, of the architecture, and this is where we, of course, suspect that there are brain areas. And to bring together all these different elements of cortical organization, brain areas, columns, cellular columns, for example, layers, but also larger scale changes like gradients, I think we will get a much more detailed understanding of the human brain in the next future. So, last but not least, I mean, neuroscience is not only a user of large-scale networks, it is also a contributor. And when you think about how ChatGPT and other large language models work, then of course we are here in a perfect situation to develop a foundation model of the cerebral cortex, also using all the rich information that you have provided here. And Christian Schiffer presented the first step of this foundational model a little bit earlier today. And then we have, when we rely on big data, we have really a lot of data. We think about several dozens of petabytes per year. This requires supercomputing. We do have it, both in Montreal and we have it also in Jülich. And the idea then would be that we really can ask specific questions to this network and ask for relationships in a very natural way. And many of you probably who are trying what ChatGPT is doing or Grok is doing, I mean, it's amazing how it's changing our way to ask questions. That's completely different as compared to what we have seen in when you just do a Google search. So we start from the atlas of the human brain, in my view, makes a lot of sense. And this is a multi-level atlas, and it means that it includes not only cortex, but also subcortex. Both is important, equally important. And we are developing these maps that include also probabilistic information, because this is a key information about brain organization. We have to consider it. But then capturing how brains differ in structure allows us to go to other data and to ask more precisely, what is the connectivity, for example, here based on diffusion, imaging, post-mortem, or in vivo. And when we have in vivo imaging of diffusion data, we can and should compare it with PLI and have a look what is, so to say, what is the fiber architecture, what is the cellular architecture. And by having them not only in the same space, but sometimes also in the same brain and the same individuals, we can, of course, ask completely new questions. It's very obvious that this is extremely storage and compute intensive. And therefore, I'm really proud that we started from the very beginning to develop a platform together where we can deal with such data. And Jupyter is the exascale computer in Europe that can be used for such projects. So we are now further ongoing with eBrains, and I will invite everybody of you join our summit. We will discuss with the community at large where we want to go to, what are the interesting questions, what is the roadmap for neuroscience, starting from Europe. And we will have a special session, and then there will be another meeting in spring where we more precisely define the contributions from the community. And for younger researchers, please do not miss the eBrain Student Conference. Also in Nice, it's also open to everybody in the world, so to say, and we would like to invite you. So that's a little bit the Europe-driven perspective, and I would like now to hand over to Ellen to build on this and bring in the broader international.
Right. Always difficult following Katrin. So Katrin talked about high-resolution brain atlasing and a little bit about AI. I'm going to talk briefly about globalization and multimodal network modeling. And as I said at the end, we would like you to really contribute your own ideas. On the globalization front, I don't need this. This is a publication that Katrin and I were involved in last year, and it was it's a visionary document on the coming decade of digital brain research. And one of the areas that I was particularly engaged in and interested in is the globalization of brain research. Increasingly, neuroscience is massively digital and massively international. You can't make predictions about brain phenotyping, genotyping in Western populations alone. You have to explore data sets and cohorts from across the world, and that's easy to say. It's not easy to do. HIBALL already has an international representation, and I think in the comments that I'm about to make, I think we are in a privileged position where we could actually address some of those problems I just described in a very practical manner. Specifically, Work Package 4 of HIBALL was charged with the problem of how do we get these infrastructures, IT infrastructures in North America and Europe to talk to each other. One of the things I should point out on this slide, some of you may mistakenly think that CBrain is a Canadian or North American version of eBrains. It's not. eBrains is a much broader international community of users and services and collaborators. CBrain is a portal to high-performance computing resources or cloud services. They have similar names, but they are quite different focus. So when we started, we had this situation that two communities on either side of the Atlantic waving to each other, not really in a position to do very, very tightly coupled interactive science together. We had non-harmonized user authentication and authorization procedures. We had different costly transfers or disallowed copying of data. This made it impossible to transfer high-volume data. We had non-interoperable HPC ecosystems, unharmonized HPC systems, application execution. Where we stand now is quite different. We have solved those issues. We have harmonization and authorization to enable CBrain portal access from either side of the Atlantic, transfer applications and tools to the data rather than the data to the tools, interoperable HPC ecosystems. I showed on the first day this as just one exemplar of a general principle of where we're at today compared to where we were perhaps 10 years ago. We are typically combining multimodal phenomic information and often genetic information. This is the example from yesterday, Turia Medina at McGill, exploring different subtypes of Alzheimer's disease trajectory, making use of imaging data, behavioral data, and various forms of omics data. Then you have, you've heard from people like Hannah Spitzer today, but also from many of our Chinese colleagues and McGill now, people are working on spatial transcriptomic data collection. Or People are developing gene expression profiles at a layer level. All of this information has to be gathered under one roof. One particular example of this general thrust is the CBIG Open Biorepository at McGill, the so-called Clinical Biological Imaging Genetic Repository. This, at this point, is the largest open tissue biorepository for brain in existence. It has over 200 cell lines, 6,000 donors, 100,000 tissue samples are made publicly available, over 100 calibrations around the world. And where we are obviously planning to take this is to combine the phenomic information on the right with all the genomic information on the left, such that in this particular example, we can explore the unique problem of rare diseases. Now, rare diseases are by definition rare. They're not, you don't have many of those people in any one city. 3 or 4 here, 4 or 5 there. If you want to do anything in that to study a rare disease, you have to aggregate data from across many, many sites. So first of all, that's a challenge by itself. It's also worth noting that rare diseases, there are 7,000 of them. So any one of them has few subjects, but there are many such circumstances. And we have to try and build an environment which allows us to accommodate such challenges. You heard from Brian Caron yesterday about CBrain. CBrain is one of the elements that allows us to do this. You've seen that it has, at this point, over 2,000 users in 59 countries, at 200 sites, over 160 complex workflows. Most of these are imaging, more relevant to the conversation that I've just been having, increasing packages exploring genomics and transcriptomics analysis. Now, this is, I think, is the important point for globalization. I'm going to get rid of all of this. What you have now is, using this infrastructure, data can be anywhere. In this example, using Eric Schulte's cell detection algorithm, the data could be in Canada. The compute can be performed in Canada. The algorithm was developed in English. The users can be anywhere in the world, and the output is accessible from anywhere in the world. This already exists. This is not a pipe dream. In another example, in this case, we're looking at the deployment of HipUnfold. This is Jordan de Kracker's hippocampal surface flattening algorithm. Again, the users can be anywhere. The data could be in English. The compute can be performed in English. It can be performed in Canada. Output visualized from anywhere. So most of these examples that I've given you are transatlantic in nature, but there's nothing specific about that particular pair. There are a number of initiatives which are ongoing around the world. One example in our own backyard is the Canadian Open Neuroscience Platform, which was conceived to allow us to share data and tools anywhere across the country, and that's been working for about 7 or 8 years now. It takes advantage of the infrastructure that I've just been describing. These are some of the open resources around the world which can take advantage of and are taking advantage of that infrastructure. I've mentioned CBIG, I mentioned the CONP. In the US, the ABCD and the Healthy Brain initiatives, these are huge multi-center initiatives, are using this infrastructure. Open Neuro makes data accessible through this infrastructure. And perhaps most importantly for this community, eBrains is also, that we've developed the compatibility between eBrains and the North American scene. Enigma is a good example, and many of you are very familiar with the Enigma situation. Enigma has been a thumping success to explore imaging genomics in over 30 different working groups. But it is worth bearing in mind that to a large degree, most of the data that is being shared there is the derived measures, for instance, the volume of the hippocampus in 100,000 subjects across the world. What we're trying to do, and we're charged with doing, is allowing us to share the large amounts of multimodal data from say, 100,000 subjects, and or leave the data where it is and then empower federated learning where the data stays where it is. You can go either way, but we have the infrastructure that allows us to deal with the raw data, not just with derived data. Another example in our experience is the Global Brain Consortium with Pedro Valdez Sosa as one of our HIBALL participants and collaborators. This is focused specifically on low and middle income countries to empower scientists in low and middle income countries. So they're not just providing data up the line to Western countries. They can ask their own questions of their own data, or indeed global data, to ask questions which are relevant to their own particular circumstance. And this is, in my view, the most important feature of the globalization move. We are democratizing neuroscience globally. Right now, as I said, the Global Brain Consortium is focused specifically on sharing tools and data in the electrophysiology space, but the principles are entirely the same if we want to apply such to imaging. And this is where I think HIBALL can play a transformative role. I'll finish with just a few comments on some of the work that we've seen at the last 2 days on multimodal networks. There are numerous examples, but this was a paper that came out last year by Ahmed Khan in Yasser Aturia's lab at McGill, which summarized various forms of multimodal network modeling. And of course, as I've been saying, the stressing point is it's not single modal. You're combining information from many different areas. You heard from Bratislav Mizik earlier in this session on the capacity to build network models across a huge variety of underpinning data types. You heard about HIPPO maps, more multimodal data that is freely available. You heard about neuromaps, multimodal data available throughout the world. You heard wonderful talk on the virtual brain and how this is combining multiple data into a common network framework. And you heard from Annie Mitjans earlier today, where she talked about her high-dimensional neuromass models. The important point here is that Annie is introducing high-dimensional distributed delay into the modeling framework. And I won't go into the details of why this is in detail important, except to say that she is using information that is derived from the literature and using biologically informed delay information and incorporating that into the model. She and Alejandro Salinas Medina, you just heard from now, they are working with the virtual brain team in Marseille with Victor Yerza to scale up both their own modeling framework and to provide resources to allow the virtual brain to handle ever larger data sets. So this is a good example of cross-Atlantic mutual support within the HIBALL framework. So I'm going to finish there and say that everything you've heard today and yesterday is really a combination of activities and disciplines from across the world, and we are here under the HIBALL umbrella. The question that we have for ourselves, Katrin and I, and now with your input, is where do we take this? Whereas there are so many ways that we can go, whether it's basic neuroanatomy, clinical translation, AI, globalization, all of these things are possible, and it's almost too rich to accommodate under one rubric. We would appreciate at this point some feedback from you about what you think is important and where we should take HIBALL over the next 5 years. The funding for this cycle may have ended, but the vision has only just begun. So I will pass it over to you. Please feel free to ask questions, and let's have a stimulating discussion for the next few minutes. Thank you.